My name is Josh Schiffman, and I'm going to talk to you about a project I've been working on with my advisor, Peter Ralph, on gene network drift and speciation. I'm actually particularly excited to talk to you about speciation, given the last speaker, Michael Torelli, because the last time I actually learned about that topic, the first time I learned about that topic in sort of depth was in his class when I was an undergrad. So, on to biology. So, what is developmental systems drift? Developmental systems drift is the idea that many different molecular pathways can produce the same phenotype. This was first articulated by True and Hag in 2001, and one of the examples in their paper was looking at two closely related species of fruit fly, Drosophila melanogaster and Drosophila simulans. Despite being different species, they actually have supposedly identical bristle pattern phenotypes as pictured here. However, if you actually cross them and form hybrids, as pictured here, the phenotype is different, which led them to ask, how is it that two closely related species can have the exact same, very similar phenotypes, yet when you hybridize them, you get a different phenotype? So this led to the conjecture that actually, despite the phenotype being the same, the molecular mechanism underpinning this trait is actually different. Since then, research in molecular evolution has revealed a tremendous amount of diversity in the genetic regulatory networks underlying lots of different traits. For example, the yeast galregulon, which is involved in galactose metabolism, is actually wired differently across many different species. Furthermore, some species actually use a completely different set of transcription factors for this regulon. Other examples that I won't go into are cell cycle control, circadian rhythm, and the gap gene network. So this leads to our sort of major question, which is, can this phenomenon, can developmental systems drift lead to speciation? We approach this with a simple, and I want to emphasize, analytical mathematical model of transcriptional regulatory networks. So, how do you model the genotype-phenotype map? Well, first, let me define phenotype. As far as the model is concerned, the phenotype is defined as the molecular time dynamics relevant to survival. So this is the concentration of molecules within the cell that are somewhat important to fitness or survival in some way. And these dynamics are the result of the specific interconnections of a gene regulatory network. For example, what we're going to be working with here is transcription factors binding promoters or other cis regulatory modules like enhancers. Also, this model also includes some sort of environmental input. So, mathematically, we assume that gene regulatory networks can be modeled as linear dynamical systems, which has two equations. Dot k of t equals ak of t plus bu of t, and then the second equation, phi of t equals ck of t. So, what does this mean? Dot k of t is the time derivative of k, which is the list of the concentration of molecules in the cell. So it's the change of the concentrations of molecules within the cell. And this is a function of the matrix A, which is the wiring of a gene regulatory network, and it's n by n dimensional. And this is also a function of u, which is the input, the environmental input, and b is some matrix that determines how the organism processes its environment. Finally, the phenotype is also a list of molecular concentrations, but sometimes it could be a subset of the molecular concentrations within the cell that are actually important or relevant to the specific phenotype. So that might have been a little bit abstract, so we're going to look at a concrete example, two-gene oscillating network. So above you see the sort of abstract general model, and below we set A actually equal to 0, 1, negative 1, 0, and it's graphically represented right here if you don't like matrices. Basically, the way we think about this is that each row of the matrix is like a regulatory region of the gene. So here you can see that gene 1, this is its regulatory region, does not regulate itself, but it is upregulated by gene 2. Again, gene 2 is downregulated by gene 1 and also doesn't regulate itself. We set B here to the vector 1, 1, which just shows that the environment can influence the concentration of both genes. Finally, C is set to 1, 0, meaning we only care about the dynamics of the concentrations of gene 1. So you can see here, there's two genes in the system, input influences them. This is their regulatory interaction. We will look at the output of gene 1. The output of that, given that the input is some sort of impulse, oscillates like this. So time, this is developmental time on the x-axis, and concentration on the y-axis. So you can kind of think of this phenotype as maybe like a simplification of cell cycle control or circadian rhythm or any sort of expression patterns that are going to oscillate periodically. So we want to connect this, like I said, to developmental systems drift. So conceptually, we kind of think about the gene regulatory network as a black box. Now, a black box takes some sort of input, like the environment, and gives you some sort of output, the phenotype. However, we don't necessarily know what the mechanism is inside the black box. Furthermore, the mechanisms, if you have two black boxes that are identical, how do you know if the mechanism inside is the same? is it actually possible? And it turns out that there actually could be, in principle, an infinite number of different mechanisms inside, and you wouldn't be able to know unless you cracked open this box. So, back to the example from before, the two-gene oscillating network. Above is the one I just showed you before. Down below is another gene network. The input and outputs are the same. So this network, instead of being 0, 1, negative 1, 0, is wired at 2, negative 1, 5, and negative 2. Despite this gene network being wired in a totally different way, its phenotype, its output is exactly the same as the other system pictured above. It oscillates in the exact same way with time. I also want to point out that in the second version, both genes self-regulate, the diagonal is not 0, and not only that, but gene 1 upregulates gene 2 here as opposed to downregulates in the other model. So the wiring is completely different. Despite this, phenotypically we can say that they're indistinguishable. So that is an example of two gene networks that do the exact same thing, that are phenotypically equivalent. Can we describe the set of all gene networks that are phenotypically equivalent? So for a two-gene oscillator, a linear gene oscillator, we can actually describe it here. This is just it in graphical form. There's one free parameter tau. So if you check any any value of tau, except for tau equals to 1, will give you a different wiring of a gene network that is phenotypically identical. So for example, the two examples I showed you before, we just had set tau to 0 for the first one and tau was set to 2 for the second one. So we expect that under constant selection pressures and a stable environment, unchanging, that evolution will explore the set of equivalent gene networks. Basically, we expect phenotype to be conserved with the mechanism to change, and we expect to see something sort of like developmental systems drift. So what are the consequences of the pattern of evolution exploring this set of equivalent networks? Do hybrids, for example, when formed between two allopatric populations, suffer from hybrid incompatibility? So before I answer that question, I should describe how we hybridize, how we cross allopatric populations. So this model is diploid. The way we make F1 or first generation hybrids is you would take two gene networks, one from each allopatric population, and just average them. To get F2 or second generation hybrids, you would take two gene networks, one from each allopatric population, randomly swap rows, so meiosis, recombined genes. That forms a gamete, then randomly grab a gamete from each, randomly grab two gametes and then average those. Then fitness can be assessed as some function of comparing the dynamics, the time dynamics, the molecular concentrations. So for example, back to that two-gene oscillator, you can see that the parental dynamics, the same one I've been showing you, oscillates with time. However, if you average the two I showed you, where we had tau of 0 and tau, so we call it A of 0 and A of 2, the F1 hybrid actually has totally different dynamics. Its phenotype is not given by sine plus cosine, it's e to the t. So we would say that that phenotype is important to survival. That would probably lead to hybrid incompatibility. So this is sort of a proof of principle. You take two gene networks that produce the exact same phenotype, hybridize them with hybrid breaks. However, you might recall that I pointed out that those networks are wired very, very differently. And we actually suspect that a change in wiring like that might not occur on the timescales we're interested in. So let's look at the hybrids between networks that are actually much more closely related. We only change the parameter of the free parameter of the gene network a little bit. So here you can see we compare hybrids, so F1 first generation, second generation hybrids, where we change the parameter starting with tau is 2, this is still the two-gene oscillator, change it by 1/100th, so 2.01, we do the experiment again at 2.1, and then we change tau to 2.5. So the bigger that number gets, the more diverged they are in genetic distance. So we plot the hybrid dynamics here. So you can see the first generation hybrids, pretty much indistinguishable. They oscillate as similar to the parents do. The F2s a cross between tau of 2 and 2.01, there is some difference. They are distinguishable, but we don't think this diversity is any more than you'd expect to see within a normal population. However, if we cross populations of tau of 2 and 2.1, so the networks change a little bit more, you can see the F1s, again, the dynamics qualitatively look more or less as they should, but the F2s start to oscillate a little bit more wildly. Finally, if we look at even larger genetic distance, where we cross 2 and 2.5, the F1s start to actually get a little bit out of phase. The F2s would oscillate even more wildly, and even one F2, this light blue line here, you can see it doesn't even oscillate at all. So, even though I've been focusing on this two-gene regulatory network, I want to point out that this model is general to any linear gene network of any finite size, so with any finite number of genes. Furthermore, all gene networks can mutually drift, and usually many directions. There's usually more than one free parameter tau. Furthermore, there is not a single there's not one phenotype with only one network architecture. You can always find a way to change the network architecture and still maintain the same phenotype in this model. And we can actually analytically describe all of them using something from control theory called the Kalman decomposition. I guess I forgot to ask the question on the slide, which is, in general, how fast do these incompatibilities appear? So we take the sort of the results and the intuition from the model I was just showing you, and we notice that the y-axis here, this is difference in phenotype between hybrids. In F1s, this is scaled at d to the distance to the 1/4, so it actually increases cortically. And even faster in F2s, the scale here is to the is quadratic to power 2. And the intuition behind this is if you picture this black line here as the neutral phenotypic space where you just move tau and moves you along the space. If you take two parental populations, say here and here, averaging them to get first generation hybrids only barely puts the hybrid off the line, whereas the F2 gets pushed a little bit further off the line. And then finally, since this line is curved, if the allopatric populations are actually more diverged, F1s actually still might be somewhat closer, close phenotypically to the parents, but the F2s will get projected down here, where this is actually phenotypically far away. So the dynamics will be very different in a system like this. So if we take these results and we plug them into a quantitative genetics model where we treat each regulatory coefficient in the matrix as a quantitative trait, we can show that actually the change in phenotypes in first generation hybrids, which we're calling delta phi, is approximately capital T over capital N, which is T is number of generations since allopatric separation, N is population size, and C is some constant. So we expect this phenotypic divergence to be linear. And then in F2s, it's the square root of capital T over N times some constant, so it would actually increase at the square root, so much, much faster. So far I've introduced a model of how to look at gene regulatory networks and their evolution, and we've been applying it to study speciation, but we think we can actually use it to study other interesting questions. For instance, are gene networks molecular Rube Goldberg machines? Rube Goldberg was a cartoonist who drew really complicated machines to do simple tasks. So far, I have not been talking about gene duplication and deletion. However, if we include them in a model like this, you can see actually that if you have gene duplication, once the gene fixes in the model, even though it might initially be redundant and unnecessary, after this rewiring occurs, you can't just pull it out without breaking the phenotype. In fact, you might not be able to pull out any genes once it gets kind of wired in there tightly. So we think this could potentially, we don't actually know yet, this is what we're working on, because maybe could lead to sort of a network ratchet where it grows in size without actually any sort of increase in changing the dynamics or complexity of the system. That's about it for my future work. In conclusion, we kind of say that there's more than one way to, if you fell asleep during this talk, there's an old saying that says there's more than one way to skin a cat. Here we sort of argue that there's more than one way to develop a cat. I think evolution will explore this space, potentially leading to the accumulation of the Jansky learning.